Let's go see if my last PhD experiment actually worked.
Yes, we are on a mission to map the brain's pain cells. So this has been done in a bunch of areas, but not the brain region I'm interested in, which is called the thalamus, which is really important for processing the negative emotional suffering aspect that comes with feeling pain. Because if you stub your toe, it doesn't just hurt. You are angry at your toe and you're angry at the thing you stubbed it on. So yeah, we're looking at those. The first stain was to tag these pain cells, and this stain is actually pretty expensive, so I'm picking up all of the ones that we used and saving it for later. And then now I'm going to add the secondary stain, which is actually going to color the cells. In this case, it's going to be a far red, which is outside of the human visible spectrum, but you can see it under the microscope. A lot of you guys asked me where I got these net transfer wells, and now that I think about it, I think my boss 3D printed these because I couldn't find them online. I found some that were kind of similar. They worked for 12 well plates, but I couldn't find any 24 well ones. So I'm going to find out his file for 3D printing and send it to whoever I can. At this point, the tissue is light sensitive, so I'm going to make sure that they're covered in foil because I don't want to mess this experiment up. This is my last one. Many of you also asked for my protocol, so this is it. You can screenshot it or send me an email and I'll send you a Word document. Once that secondary stain was over, it only took a couple of hours. I'm now going to get all my slices onto slides. So I'm filling up these dishes with PBS, which is a saltwater solution, and then I'm labeling all my slides with this Histotech pen that isn't supposed to bleed out when you get it wet, which Sharpie would. And now I take slices from each brain, put them in order from front to the back of the brain, and then get them onto these slides. I think I did 20 slides worth, and it took me 3-4 hours, but this was 150 slices. Definitely had to have some Netflix on during this because it gets boring after a while. But here are all my slices, nice and clean. And then I take this mounting medium, put it on, not too much, but not too little either. And then I'm cover slipping them with these glass cover slips, making sure to put this in an angle and then go slow and tapping it after to avoid any bubbles. Again, we don't want to mess up our images at all. At this point, I can see under the microscope if my experiment worked, but it was Friday, 7 p.m. So yeah, I'm going home. But I literally couldn't wait. So I came in on Sunday. I don't condone working on the weekends, but I just had to know if it worked. So I got my slides loaded up in the microscopes. We have 2 scanning fluorescent microscopes.
Everything should work. This protocol has worked for me very well in the past and things like that, but you just never know when it comes to science. So on it, because then it won't be it worked! Yay! Look at it. Sexy. Okay, so what we're looking at in the purple is all the cells that are activated in this part of the brain by pain. This is great. Now I'm just gonna take pictures from 20 different slides, and I'm gonna take pictures of just the whole slide, and then later I'm gonna zoom into this particular brain area, and then I'll use this AI cell counter we have in the lab to count the amount of cells here to help me build this atlas I'm working on of all of the pain active cells in this area that hasn't really been looked at before, but yay, so great. Okay, cool. Okay, cool. This is good. This is good. Okay. Thanks for watching.